Namaste to all. I'm Rani Devi from Faculty of Education and Methodology, Department of Food and Biotechnology. Today's video lecture on 19th September 2023 start in the topic poliomelitis. Poliomelitis refers to disease which caused by poliovirus, and a poliovirus is a member of Picornaviridae. It is a kind of positive single-strand RNA virus. There are three types of serotypes: first, second, and third. The route of transmission of polio is from food and water, and the virus multiplies in mucus of the throat and small intestine. From these sites, virus invades the tonsils and lymph nodes of the neck and the terminal portion of the small intestine. The symptoms of the poliomyelitis is fever, headache, sore throat, vomiting, and loss of appetite. Then after virus enter, firstly, what is the pathogenesis? And pathogenesis virus enters in bloodstream that cause anemia. In 99% cases, we can see the cases of anemia. Next, after which are very many persist and the virus enters the CNS, means central nervous system, and cause paralytic polio, which are have a higher affinity, at least 1%, and virus have higher affinity for anterior horn motor neurones of the spinal cord, and they multiplies and destroy their cells, results in motor and muscles paralysis occurs during poliomyelitis disease. The virus belongs from the family of Picornaviridae, and this genera is introvirus, and the virus type of polio is positive strand and single strand RNA. Next, there have these types of non-enveloped virus, and size about 20 or 28 to 30 nanometer. Next, after it is first isolated by Karl Steiner and Erwin Popper. There are three serotypes, and in these types, first type 1, type 2, and type 3. Which are first type 1 is Bernheim strain, and type 2 is Lansing strain, and type 3 is Leon strain. The Bernheim strain means type 1 causing moderate cause of paralysis, and type 2 serotypes of poliomyelitis virus causing greatest number of paralysis, and type 3 causing paralysis. Were minimal heterotypic immunity between serotypes, that is, immunity to one serotype does not produce significant immunity to in respect of other serotypes. Poliomyelitis virus is rapidly inactivated by heat and formaldehyde and chlorine, also including UV light or ultraviolet. As we know, poliomyelitis is a single-stranded RNA virus. So it means a virus whose genetic information consists of single-stranded RNA, that is a positive strand, which involve messenger RNA and protein. Replications of positive strand RNA viruses via negative strand intermediate. Next, after viral capsid is the protein shell of a virus. Means we can say whichever capsid of virus is a kind of protein shell which occur in poliomyelitis virus. It is consist of several oligomeric structural subunit mode of protein uh, that's known as proteomers, and which are capsid that is enclosed the genetic material of the viruses. Next, we see the pathogenesis of poliomyelitis virus, how to affect our body, and from where it enters and what's the effects. So, firstly, as we read before it, that the poliomyelitis virus enters from mouth, and their primary multiplications occur in pharynx and our gastrointestinal tract. Virus usually present in throat and in stool before the onset of illness. After 1 week of onset, number of virus in throat is less. Continue being seated in the stool for the several weeks. Next, after virus invades the local lymphoid tissues and enters in the bloodstream. They also may infect the central nervous system also, and which are replication of poliovirus in motor neuron of the anterior horn and brain, and hence results in cells destructions and causes the typical manifestations of poliomyelitis. Pathogenesis, we see the clinical features of poliomyelitis or a poliovirus. So they show some clinical features such as the incubation period of non-paralytic poliomyelitis from 3 to 6 days. Of poliomyelitis in paralytic poliomyelitis from 7 to 21 days between occurs. Response to poliovirus infection is characterized on the basis of severity of clinical preservations like infections in children and infections in adults. So, which are our infections in children of polio is asymptomatic characteristics. So, and the transmission by stool. So approximately 24% of the infection of poliomyelitis in children are measured, recorded. And the clinical preservation is known as abortic poliomyelitis and characterized by complete recovery in less than a week, low grade fever and store throat. Next, after we see the non poliomyelitic aspect, it may like a meningitis. The symptoms is stiffness to the neck and back and leg. So, in case of meningitis during poliovirus effects, we can see the symptoms like stiffness of the neck and back and legs that are usually following several days after performed, then after similar to that of minor illness. 1% to 15% of polio infections in children. These symptoms from is to 10 days at least. After pathogenesis, we studies about laboratory diagnostic of poliovirus. So, for laboratory diagnosis, we can firstly isolate the viruses. So, we isolate the viruses from pharynx, CSF, or blood through RT-PCR, and also we can perform RT-PCR. Next, we also perform serological test like from CSF and from pharynx. Serology fourfold rise in the filter systems, poliovirus infections also, and which are immunocompromised patient may have two titers actually with no antibody detect, and they still be affected with poliovirus. Next, CSF. CSF usually contains as increased number of WBCs, approximately 10 to 200 cells per mmQ. Primarily, these are the primarily lymphocytes and mainly elevate proteins from 40 to 50 mg per 100 ml. Next, after in lab diagnosis, after serological test and case history and physical parameters, we can studies on the basis of vaccines like what vaccines, what vaccines affect their during poliovirus or a poliomelitis disease occurs through poliovirus. Which are inactivated poliovirus vaccine, that is SALT. These are kinds of vaccine which are certain three serotypes of polioviruses, polio vaccine viruses, and viruses are grown in a type of monkey kidney tissue culture. Next, we also can say virus cell line, and inactivated with formaldehyde. And second polio vaccine is the oral polio vaccine, that is Sabin, Sabin. It is a kinds of trivalent oral polio vaccine set in live attenuated strain of the all three serotypes, serotype second, and serotype third of poliovirus in a ratio of 10 to 13 ratio. The vaccines are grown in monkey kidney tissue culture and varicose line culture. Then, whichever oral polio vaccines does not certain persistive, and Sabin vaccine is intestine could secretory IgA antibody. Next, the vaccine also introduced with IgM and IgG antibody in case of the rate of reversion of the Sabin polio vaccines OPV, that is leading to subsequent poliomyelitic diseases about like one case of 2.4 million doses of vaccine approximately. Next we can discuss about treatment which we give during poliovirus affected patient or poliomyelitis patient, that is because of no case of fat polio exists, the focus is on increasing comfort like speeding recovery, preventing and complications. Supportive treatments includes like pain reliefs and portable ventilators to assist breathing, and also moderate exercise. Exercise comes under physical therapy to prevent disformity of loss of muscles and functions. This is all about poliomyelitis virus, how to virus invades, and how to they occurs pathogenesis, and what the process of treatment and vaccination uh, according to vaccine prescribed by WHO. After all about complete the topic of poliomyelitis virus, like it is a kind of virus which infect mostly and cause severity, and it's multiply on mucus of throat and intestine, or we can say it's occur multiplication all over intestine and mucous membrane. symptoms so like fever, headache, and throat and vomiting. So in next class we studied about, so that's all about poliomelitis and thank you.